everyone. We're going to be starting a new unit today. It is about systematics and the phylogenetic revolution, taxonomy. Some of this information is going to be a little bit of review because we did phylogenetic trees. However, we're going to now start looking at each of the kingdoms of life, and I want to introduce you to how we do that. So, biological diversity. Biologists classify the diversity of life on Earth by identifying shared characteristics. The most effective way to classify organisms is based on the study of evolutionary relationships. So classification. It's a multi-level grouping of individuals. Aristotle was the first to classify organisms over 2,000 years ago. Eventually, groups started to be formed and referred to as genera. Singular is genus. In the Middle Ages, names began to be systematically written down using Latin. So a lot of the scientific name for animals is in Latin. Okay, so classification scheme of the Middle Ages was what we call a polynomial system. It's replaced with a binomial system by Linnaeus about 250 years ago. Polynomial, basically, it strings the Latin words and phrases containing up to 12 words. Binomial is a two-part name for each species. All right, so taxon. This is important. The groups of organisms at a particular level in a classification system is taxonomy. By convention, the first word of the binomial name is the genus and is always capitalized. The second word refers to a particular species and is not capitalized. So we're going to get into what genus and species are. But when you're asked for the binomial nomenclature, we're asking for you to tell us the genus and the species. And whenever you do that, whenever you write it down, it either needs to be italicized or underlined. And the genus needs to be capitalized, species is not capitalized. So that's our scientific name. All right, so we take a look at our taxonomic hierarchy from the smallest to the largest. So here is our smallest, the species is the smallest grouping. And as I go through and do this, you can see that you have this species of squirrel, the Caroliniasis, I don't know. It is by itself in the species. And remember, what constitutes a species is that it cannot reproduce with a different species. So species is the smallest. The next one is our genus. So these two squirrels are in the same genus together. Then you have, moving out, we have the family. Okay, so all of these organisms are in the squirrel's family. Order. Okay, so we have the rodent order. Moving out. Class. Okay, so these are all the organisms in this squirrel's class. We are going to skip subphylum because that's not important to us. Phylum is, though, however, and some of you who are taking or have taken marine biology, this should be a little bit of a review. So the phylum is larger. Then we have the kingdom, and we're going to go through the next couple weeks talking about each kingdom. And then the largest is our domain. So domain, there are 3 domains of life, which we're going to talk about, 6 kingdoms, and then those kingdoms are further divided up into phylums, which are divided up into classes, then order, then family, then genus, and then the smallest, which is species. All right, so systematics is a reconstruction and study of evolutionary relationships. We construct evolutionary tree by looking at similarities and differences between species. We now know that DNA is the best way to do that. Some problems arose because not all evolution is divergent. Some is what we call convergent. Okay, so cladistics distinguishes between ancestral from shared characteristics. Ancestral means similarity is inherited from common ancestor. Again, we just finished evolution. This is a review for you. Derived means similarity that arose within the group. All right, so we are going to talk about cladistics a little bit, and some of this, if this is a review and you feel like you know this stuff, don't write it down, okay? So an outgroup comparison is used to assign polarity. Polarize is to determine whether the particular character states are ancestral or derived. A species or group of species closely related to, but not a member of the group under study, is designated to an outgroup. Character states exhibited by the outgroup are assumed ancestral, and other states are considered derived. Okay, so a cladogram is constructed that depicts a hypothesis of evolutionary relationships. Species that share derived characters belong to a clade. A derived character shared by a clade members is a synomorphy of the clade. Again, a word we've talked about before. Each node represents a hypothetical ancestral species. Let's take a look. So, this is all too common. You guys know exactly how to do this, but I'm just going to go through anyway because it's important when we talk about taxonomy. So the trait we're looking at are jaws, lungs, amniotic membrane, hair, no tail, and bipedal. And we're taking a look here. The human is the only bipedal. Human and the gorilla have no tail. Tiger, gorilla, and human all have hair. A lizard, a tiger, gorilla, and a human, you can read this, have an amniotic membrane, so you can see how this particular cladogram is arranged based on these particular traits. Okay, plesiomorphies are what we call ancestral states. Simpliciomorphies, it's been a long time since I said these words, are shared ancestral states. Okay, homoplasies are derived characters that are lost as species in the clade re-evolve to the ancestral state. Principle of parsimony favors the hypothesis that requires the fewest assumptions. So that's always true in science. It's reoccurring. So we favor the hypothesis that requires the fewest assumptions. So systematics and classification. This is something new. A monophyletic group includes the most recent common ancestor of the group and all of its descendants. So that's what we've been dealing with up till now. With a cladogram and making a cladogram. By definition, a clade is a monophyletic group. So we have a paraphyletic group, and this includes the most recent common ancestor of the group, but not all of its descendants. Polyphyletic group does not include the most recent common ancestor of all the members. So we're looking at here, we have a monophyletic group. Okay, here's a clade. Here's a paraphyletic group. Normally looking here. And an example of a polyphyletic group. So analogy versus homology, some important differences here. Examination of the characters on a cladogram can provide insight to how they evolved, how many times they evolved, and how complex characters evolved. So homologous traits, we know this, similar traits in different clades are shared with a common ancestor. Analogous traits, which we haven't talked about before, are similar traits that have evolved independently in different clades. So there's a big difference there. So we're looking at a cladogram here, arms longer than legs, long feathers, long arms, highly mobile wrists, downy feathers, wishbone. So when we look up the clade, we're saying light bones here. So dinosaurs did not have light bones evolved here, and then all of these organisms have light bones. All right, so let's talk about the kingdoms of life, and the writing small, which means I crammed a lot of stuff in here. So most biologists used a six-kingdom system. We have Animalia. We go through every one of these kingdoms separately, so this is something we're going to be vague with. It contains only organisms that are multicellular and have no cell wall. Plants contain only organisms that are multicellular, have a cell wall, and are photosynthetic. Fungi are multicellular organisms and single-celled yeast. Fungi are very interesting. You're going to be surprised to learn the things that you learn about them. Protists are unicellular and eukaryotic. Archaeobacteria are very diverse. They include methanogens and extreme thermophiles. They're prokaryotic. But we also have bacteria, which are other prokaryotes, but these two actually have their own domain, which we're going to get to because they're so diverse and so different from each other. So domains, here we are. Domain's the largest taxonomic group. So there's 3 domains. So we fit all of these kingdoms into these 3 domain systems. We have bacteria, archaeobacteria, and eukarya. So bacteria here has its own domain and kingdom. You're probably asking why. Because there's so many of them, it's impossible to ignore that the majority of life on Earth is bacteria. And then, I didn't mean to do that. Sorry. And then we go back, Archaeobacteria has its own domain. And then Eukarya includes animal, plant, fungus, and protist, all grouped together in one domain. So we can just take a look. And it's interesting, when I first started teaching, they had just come out with this new domain system, so I didn't learn this in high school. I know you probably don't care. But I just find it interesting when I learned about it. So here's our domain system. Remember, domain is the largest classification system with bacteria, archaeobacteria, and then all of these fall under eukaryotes. All right, so let's take a quick look at archaea. They contain some key characteristics. Their cell walls lack peptidoglycan, and that's an important difference between archaea and regular bacteria. They possess unusual lipids and ribosomal RNA, so very unique to their own domain. There's 3 general categories of these archaea. One is methanogens. They obtain energy using hydrogen gas to reduce carbon dioxide and methane gas. You could find these guys at the bottom of the ocean in the deep sea vents. And if you took marine bio last semester, we actually watched a video talking about those. Then we have our extremophiles, the second group. They grow under extreme conditions. Okay, there's thermophiles, there's halophiles, and there's pH tolerant. So these guys live on the brink of extremely harsh conditions. There's pressure tolerant. And then we have the non-extreme archaeobacteria that grow in the same environment as eubacteria. So this domain, archaeobacteria, since we're just finishing evolution, and we know that the first cells were protobionts, well, we are hypothesizing protobionts, and then we go to prokaryote. We're thinking that the first prokaryote was this archaea domain, because if you think about how the Earth's crust was, how the atmosphere of the Earth, it was had a lot of methane, a lot of CO2, no oxygen, very hot, a lot of extreme conditions. It is almost widely accepted that the domain archaea was the first cell on Earth. Again, something we just found out recently. So we're taking a look at the tree of life. Here's our archaea. We have our common ancestor down here. And then we broke off into these 3 different domains. So we're looking at the different types of archaea. And again, this is their genus name. It's capitalized. All right, and you can see the word coccus. Coccus, we remember that means circle. All right, and then we branch off to our regular bacteria. And then we branch off to eukarya, which means along the way, some cells started eating mitochondria and chloroplast. I don't know what just happened there. So then let's go to our bacteria. They're the most abundant organisms on Earth currently. The most successful evolutionarily wise. Most taxonomists recognize 12 to 15 major groups of bacteria. They're as different from archaeobacteria as they are from eukaryotes. So that's why these guys are separated. We have eukarya. They appear in our fossil record about 1.5 billion years ago. Again, we think this is because of endosymbiosis and the eating of the mitochondria and the chloroplast. They have a very complex cellular organization. We spent a whole semester talking about eukaryotic cells and what goes on inside of them. Fungi, plants, and animals are well-defined evolutionary groups. They're largely multicellular, and the diversity among protists is much greater than or within between fungi, plants, and animals. So again, we spend a whole day talking about protists, and I'm hoping to actually get out into a lake to find you some samples of protists so you can look at them under the microscope. Just got to warm up a little bit first. So eukaryotic cells, we know this, guys. It's just a reminder for you. If you know this and you don't need to write it down, don't write it down. Mitochondria, and they have a mitochondria and chloroplast. We know mitochondria and chloroplast have their own DNA. Some protist phyla have also acquired chloroplast and are photosynthetic. Mitochondria and chloroplast are both, Anyway, moving on. So the key characteristics, compartmentalization of their organelles. Okay. They're multicellular. Excellent. Let's keep going. They alternate between this is always hard for me, syngamy and meiosis, which is a union of male and female gametes. So basically sexual reproduction. Okay. So here's a diagram of the evolutionary relationships among all of the 6 kingdoms, and I always find this really interesting. So we look at our original cell, and we have our archaeobacteria and our bacteria. So what I want you to recognize, okay, is where eukaryotes come. Eukaryotes, it seems, are more closely related to archaeobacteria than our regular bacteria. So at some point, eukaryotic cells break off from archaeobacteria, and we go here, and you see we have a little connection here with fungi. So there seems to be a little bit of wavering on what exactly we think happened. Again, we've been looking at diagrams not based on DNA. Okay? So we're just looking at features of the domains of life. You have archaeobacteria, amino acid that initiates protein synthesis, methionine. Great. Start. Formal methionine. So our start codon. Our introns. So archaea has some introns. Bacteria has none. And eukarya has introns. So membrane-bound organelles, absent, present. It's worth looking at to see how things are related. So a nuclear envelope, number of RNA polymerases. We see a lot of similarities between archaea and eukarya. Okay. No peptidoglycan in the cell wall. Ooh, none here, some here. Response to antibiotics, growth inhibited, growth not inhibited. That's a big deal. Okay. So then again, we're just taking a look here at the 6 different kingdoms. This may come in useful later on when we go through and talk about each one of these kingdoms individually. Here we go again. All right, so viruses is our next topic, but I do want to just talk about them for a minute. You learned them last year with me, but again, you didn't learn them with me because I wasn't in. So we spend a day on viruses because they're so important. They don't satisfy the basic criteria of life, so they're not living. They can't reproduce on their own, and they cannot be classified as living. So we cannot logically be placed in any of these kingdoms because of their characteristics. So the question arises, should we have a new category for viruses? Well, no, they're not living because they can't reproduce on their own. They need a host cell. So what they appear to be are just fragments of nucleic acids originally derived from the genome of a living cell, which is really interesting. And another interesting disease-causing material is something called a prion. Prion is something we should talk about when we get into viruses, but it's not a virus. They infect organisms at all the taxonomical levels. So every kingdom of life can be infected by a virus. So we don't need to write this down. There you go. There's a picture of a koala for you. I'm trying to pause this, but it's not letting me. Doesn't work.